All right, so my name is Lavrency Jovanska. You can call me Lala. I'm a senior marketing specialist at Taiwan Advanced Nanotech or TANBead. And today in this webinar, we will be discussing about forensic DNA analysis and applications. Now, this webinar will be comprised of 4 parts. The first part is nucleic acid extraction, and from the second part, we will be focusing on forensic science, specifically from scene to lab process. So we'll be looking at the different types of samples that we can collect and how to process them. From the third part, we'll be looking at the applications of forensic science before finally concluding the presentation. After the conclusion, we will have some time dedicated for question and answers. But if you have any questions along the way, please feel free to type your questions into the chat room. Okay, so we'll be starting with the first part, the nucleic acid extraction. So before moving on to any DNA analysis, it is crucial that we have DNA of high quality so that we will minimize any problems that might occur on the downstream processings. Generally, the extraction methods are divided into manual preparations and automatic preparations. For manual preparations, there are chemical-based and column-based extractions, and as the name suggested, if you're working with manual preparations, there will be a lot of manual pipetting along every step for every sample. The bright side is that we'll be able to customize any step if necessary. Now, for automated extractions, what we will do is that we will put the samples into a reagent kit and then plug the reagent kit into an instrument, and that instrument will work the extraction process for us. Now, the instrument might use up and down mixing or spin mixing. If you're familiar with TANBead instruments, we are using spin mixing in order to increase the processing volume and also to minimize cross-contamination. Now, we are offering different instruments with spin mixing depending on the number of samples that you're working with. So if you're working in a lab for a small amount of samples, probably like 1 to 8 samples at a time, then we would recommend Maelstrom 8 auto stage. For a larger number of samples, probably 1 to 48, then Maelstrom 4810 will be more suitable for you. Or if you're working with a large number of samples, probably 1 to 96 at a time, then we would recommend Maelstrom 9610 for you to work with. Aside from instruments, we're also offering the reagent kits, so you don't have to worry about the instrument and reagent kits compatibility. Depending on the sample type that you're working with and your target nucleic acid, we have a range of kits that you can select from. So let's say you want to isolate blood DNA or blood RNA, or probably plant DNA or plant RNA, or both DNA and RNA of virus. But today we are going to specifically look at forensics DNA. Now, this is the installation map of our products. So TANBead products has been distributed to more than 80 countries worldwide. So I believe it will be easy for you to get our products. But if you're having any difficulties, then please contact us and we will help you get connected. So that was briefly about the nucleic acid extraction part. Now we're going to look at the forensic science scene to lab process. Now, what makes forensic science different from others is that it is working closely with the law and justice system. So let's say we're working with the police to track and apprehend a criminal. We will be collecting samples from crime scenes, and the samples might be challenging because We're probably not really sure where or from whom the samples are coming from, so they are of question or are unknown owner. And the quantity might not be much. Depending on the environment, they might even be degraded, and they might contain some contaminants or inhibitors. And since we are trying to get as much DNA as possible from that crime scene, so we might be including some unusual ones. Probably we'll be looking at chewing gums or cigarette butts from the crime scenes, or maybe nail and hairs that we found on the floor or on the walls of a crime scene. And we will also be collecting DNA from witnesses or suspects, probably in the form of a buccal swab or a blood stain, semen stains, or others. For easier imagination, we're going to have a mock case. So, this is a crime scene. We have a victim and a few witnesses or suspects. The police would have a few questions to solve, probably when the time of crime was committed, how the crime was committed, who would be more likely to commit the crimes. So after looking at the questions and posing more detailed questions, we would come to a question like, who among these witnesses are more likely to commit the crime, and who among them had been to the crime scene? Now then, our police team might collect some cigarette butts, hair, dried blood, or nail from the crime scenes, which they are not sure from whom the samples are coming from. They will also collect DNA or samples from sources that they are sure of. So maybe they will be collecting the blood of the victim or his semen stain or hairs. From the witnesses, they will also collect DNA samples in form of buccal swabs, nail hairs, or chewing gums, or other things that they think would be necessary for the investigations. Now, after collecting all these known and unknown samples, we will treat them, extract them, and analyze them. Now, to be sure that we are working with DNA of high quality that will not cause problem on the further downstream processings, we'll be extracting the samples with 3 different instruments and the respective reagent kits. And we will analyze all of them in order to make sure that our nucleic acid is okay. Now, since we are looking at a crime scene, probably there are not much samples to work with. When using TANBead forensic kits, here are the minimum suggested amount of samples that we can use in order to collect enough DNA for downstream processings. So when you're using TANBead forensic kits, just 10 microliter of blood stains in a swab would be enough for you to have enough DNA for the further processings, or probably just 10 milligrams of chewing gum. Or if we have fresh blood collected on FTA or filler papers, then later on we will just need to cut a circle, probably 6 millimeter in diameter. And after extracting, then we will have enough DNA for the processings. Now, for this group of samples on top here, we'll immerse them in incubation buffer and protein escape for enzymatic digestion. This immersed sample would be incubated at 56 degrees Celsius on a shaker rotating at 900 RPM for 1 hour. And then we will transfer all the lysates into the reagent kits. Now, for the group of samples on the bottom, we will add incubation buffer, protein escape, and DTO3-YOL, or DTT. Now, DTT is used to break disulfide bonds. So, samples such as semen stain, will contain a lot of disulfide bonds on the sperm cell coating. While for nail and hair will contain a lot of disulfide bonds because they contain keratins. So first we would have to break these bonds before we can extract out the DNA. And then the following incubation steps are the same as the first group, in which it will be incubated at 56 degrees Celsius on a shaker rotating at 900 RPM for an hour, and then we will transfer all the lysates into the reagent kits. Now, these reagent kits will then be plugged into our instruments, and after 60 to 70 minutes, we will get the DNA all extracted and ready for further processings. The first downstream processing we'll be looking at is qPCR analysis. Now, in these two graphs, the genes that we amplified are GAPDH and actin-B because they are the housekeeping genes. So we just want to make sure that we are getting enough DNA in our elutions. Now, here we are working with 8 different types of samples: blood stain, dried blood spot, semen stain, nails, and others. For each of the samples, we extract them in 3 different instruments and their respective reagent kits. Each of them are replicated 3 times. So now that we can look at these graphs, we can see how the CT values of all the samples extracted on different instruments and different reagent kits produce the same CT values. So it means that the kit we're working with, the instruments we're working with are stable and replicable. So we're not seeing any problems on the qPCR analysis. So we will evaluate it further with gel electrophoresis. Now, the instruments we are using are SLA E13200 with the 6TF kit. Now, the second instrument is Maelstrom 4810 with the M6TF kit. And the third instrument is Maelstrom 9610 with the kit W8TF. Now, for the gel electrophoresis, the one that we place in as the sample are specifically mitochondrial DNA from the 8 samples mentioned before. Now, regardless of the type of instruments, kits, or samples, in here we're seeing that all the bands are very clear. We are not seeing any smearing, and the bands are really tight. So we can see that this mitochondrial DNA is sufficient for further processings. Now, the next one, mitochondrial DNA analysis, is the heart of this forensic analysis we're going to cover today. Actually, for forensic analysis, aside from mitochondrial DNA, we can also work with nuclear DNA or Y chromosome DNA, depending on the case. So if we have a lot of samples to work with, then maybe we can work with nuclear DNA to do DNA fingerprinting. That way we will have more informations to dig from. Or if we are working on a sexual assault case where it is where we are really sure that the perpetrator is a male and the victim is a female, and in our samples there are a lot more of the female DNA than the target male DNA, then it is suggested that we use Y chromosome DNA analysis. Now, in our mock case here, in our murder case, we're not sure whether or not the murderer is a male or a female. And we also don't have a lot of samples to work with. So if we want to obtain nuclear DNA, it might be hard to do so because for nuclear DNA, there are only 2 copies available per cells. Meanwhile, for mitochondrial DNA, we might be looking at hundreds to 1,000 mitochondria per cell. And in each of these mitochondria, there are 2 to 10 copies of DNA. So in one cell, we will be looking at 2,000 to 10,000 copies of DNA. And also because mitochondrial DNA are maternally inherited. So in the cases where the witnesses or the suspects are not available, let's say they went missing or had passed away, we would still be able to obtain their mitochondrial DNA from any members of their maternal lineage. Here we are looking at the picture of the mitochondrial DNA. So it is comprised of around 16,000 nucleotides and encodes 37 genes, including 13 essential polypeptides, 2 ribosomal RNAs and tRNAs, and the D-loop region. Now the D-loop region here is what we are going to look at because it contains 3 hypervariable regions, termed HV1, 2, and 3. So as the name suggested, these regions are highly polymorphic, so it could be mutated easily, and it will vary from one individual to another unless they are maternally related. If they are not related, then the chances of 2 individuals having the same HV1, 2, and 3 sequences could be lower than 1%. That's why mitochondrial DNA is also used for genetic diversity and population origin studies. Now, in our case, we're going to sequence this mitochondrial DNA and compare the sequences between the samples. So we have 8 samples, and we sequence the HV1 and HV2 of the mitochondrial DNA from each of the samples. So the original sequence would be a lot longer, but in this table we have comprises the certain spots where they are different from one to another. Now in this table we can see that the first 4 samples, blood stain, red blood spot, semen stain, and nail, have the same sequence, so they came from the same donor. Let's call it donor 1. While the 5th sample, hair follicles, have different sequences compared to the nail samples, so it's coming from a different donor that is present at the crime scene. And it matches the buccal swab sample which came from one of the witnesses. So now we have 2 different donors. Let's call it donor 1 and donor 2. Now for the cigarette butts and chewing gums, it have different sequences from both the hair follicles and nail samples. So it's also another different donor. So now we have 3 donors in total. Now in this slide, we're just looking at the mitochondrial DNA sequence from sample extracted using SLA E13200. This slide, we're looking at the exact same samples extracted with mouse from 4810. And the result of the analysis are exactly the same as the SLA instrument. So now we will also compare it to the result of extraction using mouse from 9610, which also produces the same result. So in here we can be very sure that extraction using different types of TANBead instruments and kits will produce results that are stable and replicable. Alright, now that we know there are 3 different donors from the samples, it's time to match the samples to their donors. So we have some samples in which we are sure who the donor are. Let's say the blood stain belongs to the victim for sure. And in the crime scene, we have nail samples in which we are not sure who the donor was. Since the sequence matched with the blood stain, it means that the nail belongs to the victim. Now, for the hair follicles we found on the crime scene, it matches the sequence of buccal swab from one of the witnesses. So this witness, let's call him witness A, was present at the crime scene. Also goes for cigarette butts, which matches the sequence of chewing gum from one of the witnesses. So here we have witness B also present at the crime scene. So now that we can conclude there are at least 3 people present at the crime scene, we can give this evidence to the police and they will compile it with all their different evidences, maybe witness interviews or recordings, and finally they will be able to recreate the whole story of the crime. Now we will be moving to the third section, which is forensic science applications. Here I divided the applications into 3 general groups, with the first one being crime scene evidence analysis, just like the murder case that we saw before. But actually, one of the most common application of forensic science is to solve sexual assault cases. And it can also be used to solve burglary cases. On the second category, we have victim identification. So we might identify victims of war, accidents like plane crash, or natural disaster like earthquakes or tsunamis. On the last group, so forensic science can also be used for parentage tests like paternity tests or ancestry tracing. We will be looking at all of these applications in more detail, with the first one being crime scene evidence analysis. So I said before that one of the most common applications is to solve sexual assault cases. In this type of cases, both the police and the victims could request for an examination. You might Google rape kit or sexual assault evidence kit, and it will refer to a series of hospital or police examination that a victim can go through in order to collect evidences of sexual assault, such as hair, semen, saliva, or blood, and they will compile it into this box, the rape kit box, and then the police will send this to the forensics for further analysis before finally delivering judgment. And the second type of crime, like we saw today, the murder case, forensic science can be used to investigate both ongoing cases and cold cases. Cold cases is referring to a case that has not been solved, even years after it had been committed. So even when the suspects are missing or had passed away, we would still be able to work his or her case if we have workable DNA, or we can collect DNA from their surviving family members. Usually we'll be looking for cigarette butts, blood on or hair in these cases. Now for burglary cases, we'll be specifically looking at the break and entry area of a house, like broken windows or doors, because in other parts of the house, we will be more likely to find the DNA of the house owners themselves. So during the entry or escape, we are more likely to find the hair or blood of the perpetrator. In the second category, victim identification, we might be looking at victim of wars. It might be the soldiers, which is not recognizable by other means. So probably we'll be looking at the remains like just an arm or just a leg of a uniformed soldiers, and that's when we really need forensic science. Or in another case, we might be looking at the civilians which were attacked by the soldiers. So even in war situations, soldiers are not supposed to attack civilians, and they might be charged for war crimes. So we might be looking at the bodies of civilians and search for the DNA remains of the soldiers that attacked them in order to charge war crimes against these particular soldiers. For victims of accidents like plane crashes, we might be working with victims that are highly carbonized or burned. So They might be unrecognizable through other means. There might be just very little part of them that are remains that could be extracted. But thankfully, because plane passengers are well recorded, more than 25% of airline accident victims were confirmed through DNA profiling. Of course, the number would differ from case to case, especially if they crashed into a residential area. Now, if they do crash into a residential area, then we'll be looking at them like victims of disasters. So victims of disasters like earthquake, landslide, or tsunami, they might be buried on earth for a few days, or they might be drowned on water or other environmental conditions that might accelerate the body decomposition. And especially if it affected a large area, then we might have to match the DNA of fellow victims because there might be no surviving family members that would come to claim their bodies. So we might have to match their DNA and written records, probably like residential ownership records, in order to identify who these victims are. Now, on the last group, forensic science can also be used for parentage testing, such as paternity tests. So, a court could order a paternity test for legal purposes, like child support. One of the most common cases is when an unmarried couple splits, and maybe the mother would be asking the father for child support, for financial supports. Now, because we will be looking at 19 to 18 years of financial responsibility, in addition to the rights of visiting the child, and also the rights to child custody, the court will first issue an order of paternity test before delivering their judgment. Now, for changing birth certificate, paternity test is also required because then it will be related to the rights child of inheritance or veteran benefits or other things that a parent can pass to their children. Paternity test might also be required for applying immigration visa because then it will be related to the right to citizenship, the rights to health insurance, or retirement pensions, depending on the country's regulation. Now, if paternity test can be conducted when both the parent and the child are present, it could also be conducted even when the father is not available. So, if the father had passed away or had been missing, then we could construct the DNA of the missing parent using the samples from his or her surviving family members. That way we will still be able to confirm the paternity. Now, aside from paternity tests, which is looking at a close relationship between a parent and a child, we could go even further up the family tree. We can be looking at ancestry tracing. So if you're familiar with websites like ancestry.com or 23andMe, they are offering DNA analysis, and they will compare this analysis with written records to understand the client's family lineage. So a client might send them saliva samples, and this company would analyze the DNA on that saliva and will send this client some written report stating that, oh, you are likely 70% Asian, maybe, or like 20% European, 5% Jewish, etc. And they would also connect this particular client to other clients of theirs that are likely to come from the same family. So then they together can discover their medical history, prevented inherited diseases to take place, or just meet up and have a good time or something. Now, these are the TANBead forensic kits, which application we have discussed today. Now it's time to conclude the presentation. So today we have looked at how nucleic acids are highly used in molecular research and diagnosis, particularly in the field of forensic. And we also had looked at how TANBead can provide high quality and quantity of extracted nucleic acids, especially when coupled with the automated extraction system. And when you do choose TANBead, we will send you products of assured quality. So that is the end of the presentation for today. Now we're going to move into the question and answer section. If you have any questions, please type in your questions into the chat room.